Hi everyone. So I don't often do live streams, so I thought, and this was kind of the only time I could do it. And so if you hear sound in the background, I put in a load of enrichment, and the guinea pigs really make a lot of enjoyment out of that. So anyway, I kind of planned half a video. Oh, just gonna check that it's working, because let's see. This hasn't gone as well to plan as I thought. Oi, stop chewing. Okay, let's see. Live. Let's check. There we go. So it's saying on one thing that my camera's not working, and on the other it is. So it's a little bit confusing. Good. So I think, so I kind of planned it kind of because I made the thumbnail and it took a while and it was a bit of a generic thumbnail. So I thought I'd talk a lot about, well, just fish biology in general and its importance, why it's interesting, like case studies and stuff like that. But I've got a lot of flexibility, just because I kind of planned it as partially a video, partially a live stream, depends on what happens, because obviously major clash in sort of schedules. So I think most of the people viewing probably know who I am, but obviously there's going to be quite a few people that don't. So my name is Rebecca. I'm a fish biologist and an ichthyologist. I study evolution of catfishes. So I'm a PhD student, and that's my kind of topic. And I'm looking at how their morphology changes over time. Or changes over time, but also between the different groups. So what else have I done? I have actually done research on corals, sadly, but I did do a master's project on them. I've also done a little bit on Callichthyidae. So it's really warm in here because the fish tanks heat it up, and then this house is like south facing, apart from there. But yeah, so I did a little bit of work on Corridors and Callichthyidae. Obviously, I've got the papers published on Corridors, but I actually specialise in Loricariid mostly. So my background is kind of in that, other than I did work for the aquarium trade. I now say 7 years because previously I worked in the trade side of it, but now I work in the sort of scientific laboratory side of fishes as a kind of a side thing. So I do a lot of different things. So that's kind of what I do, what I'm about. Anyway, so a few people have joined. Hi, Mickey, and hi, Brian. Thank you for joining. And it's good that you can hear me and see me, because it's always difficult to know whether it's working or not. So a lot of people, like, fish biology is quite a difficult topic to define, or because obviously ichthyology doesn't help when people see it. Because ichthyology is basically fish biology. When people see and think about the word ichthyology, they're first thinking of ichthyosaurs, which are marine reptiles. They get called dinosaurs, but they're not. Ichthy meaning fish. I think it's Greek. So ichthyology is the study of fish. But the problem is, what are fish? And so fishes is what's known as a polyphyletic group, yes, it'd be polyphyletic. So it includes one like ancestor, but not all of the descendants. Is that paraphyletic? That'd be paraphyletic. But they're such simple words. But this makes it quite difficult because it's kind of, it starts with, so it starts with a gnatha, which is the jawless fishes. So they're like your hagfish and your lamprey. The only difficult bit with this sort of idea is that so the only difficult thing with defining fishes is that, as far as I remember or know, the hagfishes aren't vertebrates. So you can't say all fishes are vertebrates because they don't have a vertebral column, I believe. And then there's some issues with as lamprey develop. I think they don't have a true cranium or something. So it's really difficult because there's terms that kind of were used, but they don't count for all fishes. So fishes is kind of all the way from the jawless fishes to, I guess, most of sarca. And yes, this is the problem. Every vertebrate is technically a fish because Sarcopterygi, which is lobe-fin fishes, contains tetrapods. So generally, when defining fishes, it's so difficult. It's because it's like every sort of vertebrate, but tetrapods. So that's amphibians, reptiles, mammals, birds, but then also hagfishes in a way. And then you can't just say it's aquatic because dolphins, but there's loads of old depictions of Monty, stop chewing. He's going to town with this bit of cardboard I put in to line their litter tray. But so it's really difficult. And there's loads of actual groups that are part of fishes. And in the aquarium trade, in the aquarium hobby, we only deal with what's known as like actinopterygi. So that's your ray-finned fishes. Now there are exceptions. Some people keep rays and they're known as chondrichthyes. So it's kind of this branching. So a lot of the names we commonly use don't actually have equivalents we commonly use. So the equivalent for the jawless fishes would be jawed fishes, which is gonathostomes, but no one really talks about gonathostomes. And then the comparison to chondrichthyes is osteichthyes, chondrichthyes being cartilaginous fish, osteichthyes being bony fish. And then osteichthyes narrows down further to that Sarcopterygi, actinopterygi. And I think those are the two. But we mostly deal with what's known as teleost fish, actinopterygi. And yes, hagfish are extremely creepy. They also release a lot of like slime. And it's quite interesting if you look them up. They actually tie themselves in knots around when they're in their own slime just to try and get it off. But sometimes they do suffocate in the slime, I believe. But yeah, they're one of the sort of most oldest groups, or the most similar to what the oldest vertebrates would be. And they've been around a long time. But there has been some evolution, I guess, a bit like with lamprey. But that's what the sort of ancient groups of fishes would have looked like. But there's, yes, definitely a lot of names. And because I kind of want this to be an audio format, I don't really want to, I was going to make a load of slides, but then I kind of want to just test entirely audio format in a way. But teleost fish are the main ones we deal with. That's stuff like sort of tetra, catfishes, all of that. Main fishes we deal with, apart from if you keep obviously stingray, then they're cartilaginous fishes, and they place outwards with like sharks and stuff. Sharks and yeah. And I believe so. I did look at them quite a bit in my undergrad, and some of those fossils are a bit vague, but I can't remember. I think it was the Gnathans, but I try and avoid fossils. There aren't many actual fossils for Loricariids, probably just due to locality. So them being what I study, they're the fossils I've looked at, and they're very sort of vague, usually like one bone. And there's kind of like a lot of fishes maybe don't fossilize so well, or the radiation of fishes makes it difficult to compare, because I think for mammals and that lot, we have a much further understanding of fossil species, whereas for fishes, I don't think we do. Like, we have like datings on like catfish when they first evolved, but it changes, like especially for Loricariid, so plecos, they evolved Well, we thought they evolved age of mammals, early Miocene, then the era before. And then there's an African Loricariid, which totally throws that out by, I think, quite a few million years. But yes, and that's why, so I listen to a lot of audio formats, like podcasts and stuff, because I like to do other things. And unless I have multiple screens up, I don't like having, I don't like just, what's it like, having one thing to watch. I like to do loads of things. That's the name because, yeah, so I prefer, so if I wanted to put on Spotify, I could, because that's why I listen to a lot, because YouTube, I think when I close the screen down, it'll pause it, and I'm not going to walk around with my screen open on my phone. And so then this is the name of the African Loricariid, Aphocasada. I know that the second name, I believe, is like either Portuguese, Spanish for what a lot of people call plecos. So that's where the name originates, and it makes sense. It's quite a logical name. And interestingly, so Feather Fin Films has brought up that he thinks, she thinks a lot of loricariids would have done well in Africa. And I would say potentially yes, and there seems to be a gap in when it comes to rasping fishes. There's so much diversity of loricariids in South America and Central America. Yet in Africa, they seem to be like dual purpose fishes and not so specialized. Like you've got Synodontis, and there's not many of them really rasp. Then you've got Hyloglanis, which we don't see in the aquarium trade. And they are riverine as well, which like Loricariids are. Well, Synodontis, obviously you've got the lake synnos, but a lot of them are riverine and rasping, but not like to the extent of Loricariids, and not that diversity. So maybe there might be something in it, and I think it was mentioned in the paper, but me and biogeography, I am not the best at biogeography at all. So especially when it comes to all the different plates of South America and stuff like that. Yes. Yeah, so the only rasping fish are the mochicids. So that's Synodontis, Chyloglanis, other glanis fishes usually. But not many of them, like some of them, they all seem to be able to do other feeding niches as well as they feed on algae in a way. Like Synodontis bichardi, really elongate, and it has that rasping mouth, but I can see them eat on earthworms as well as they would ever feed on algae. I don't know if those fish were wild caught. They could have been hybrids, which did happen when I ordered in Shoutedeni. Everyone will think of the puffer Shoutedeni, but this is the Synodontis Shoutedeni, and they might have been, yeah. So, going to be showing a lot of these comments, but yes, I think there's a lot more like, they do have a lot more flexibility there, or diversity of flexibility. It's really difficult to explain because when I look at more just the bone shape itself, but there's also their secondary pairs of jaws that haven't really been studied, but are very popular to study in African cichlids. Not studied, I believe, at all in Synodontis, other than a vague mention of the secondary pairs of jaws. But I've got 3D printed Loricariid jaws. So this is Lichthosis. So this is from Lichthosis lichthoides. So I didn't get one more common to 3D print, but it's just why I quickly got on the computer at the time and 3D printed it. And it has broken a bit because 3D prints are always delicate. But you can see the lower jaws here. So that's the dentary, and these ones will be a lot more flexible, especially in this fish. So this fish, I have an image of its actual teeth that I put under microscope, and I swear they weren't as flat. So maybe it was the 3D printing error, or it could have been an issue with how I was doing it. But you can see here, that's how the lower jaws are, and they've got quite the amount of replacement of teeth, but that's for the bottom one. I only printed out one of the top. So these will be much closely connected, and they have a lot less mobility. It's also where the CT scan cut it. I don't have the best. It's kind of cut in half, but there's something that definitely. But I've never actually really looked at the jaws of Synodontis. I don't think they're definitely not as diverse. They're more probably similar to, yes, as mentioned, where is it? The Astroblepus, which no one ever gets Astroblepus into the hobby. They're weird things. The one ICT scanned looked absolutely it looked like a turd. So the photo I was like, oh my God, it looks a bit crazy. But so what was I going to say? Yeah, Astroblepus is a little bit more generic. And which one did I manage to get a scan of? Not Delta-A, Lithogenae, which is another one you won't see in the trade. These are like highly riverine fishes, very faster flow than even like some Ancestris or Ketostome. I don't even think they're found together. Okay, so did you get them to? So Featherfin Film says that he's owned Astroblepus. Were they from the aquarium trade or anything? Or I don't even know how easy they'd be to fish. I know might be some issues with like catch locality to be able to get them out. There are a few like actual Loricariids at very high flow waters, but you never see them in the trade. But there's a lot of Loricariids. Like one of my favourites is I always forget its name, but it's one of my favourites. It's actually related to Otocinclus, but looks entirely different. But the spelling of it is just constantly, I have to double check for spelling errors. But so getting a little bit more onto topic, I guess, for now. When like thinking about fish biology and how we're going to apply it to fish keeping and understanding it, is knowing where to research. And this is really difficult. So I always go straight to the scientific literature. So I'll just I always go straight to scientific literature and also theses, which are peer reviewed, but you obviously need to include some scientific, need to critically analyze. But the problem is they're not always the easiest to access. So, if I get... Sorry, so I'm just looking... So, Featherfin Films has just said, is it Hyperptoma? No, it looks completely different. I believe they're actually, this genus that I really like is restricted for export. So it's a Neoplecostomus, I think is the clade that you can call it now. But it used to be known as, in the group, Neoplecostominae. Pariahapsis. You won't see them in the aquarium trade. I believe they're heavily restricted. But I really like them. And the Neoplecostomus. Yeah, I think that's... Yeah, and as... Featherfin Films has mentioned that it's the oxygen, the flow. Houses might even just be too warm for them. The oxygen levels might just be too low in general. A lot of the fish that we keep might be high flow, but they're also happy to deal with very high temperatures and much higher than we give them, like Ryojingu temperatures, even Orinoco. Like I had to double check some of these. So it's always worth double checking or fact checking your own information just in case, because there's sometimes is things that get missed. And I double checked Rio Orinoco, that's like usually above 30. Oxygen's going to be really low in summer if you kept them in that at the highest or sort of higher end of their natural temperatures. And then Rio Namuata, where Ancistrus dolichopteris is found, or Dolopterus, I think is how a lot of people pronounce it. And they're found well above 30 as well. And even so house temperatures, even in the UK in the summer, are too warm, I think. Our natives can't even cope with, or most of our natives really don't like it. I just would like to keep them. There's some fish I just really want, like a Chiloglanis. Hopefully, maybe one day I can get them. Just, I like challenging fish. But yeah, I think, Texas definitely might be a bit warm. And chillers, we don't use them in the freshwater hobby at all, really. They do exist for marines, but I get in the marine side, I'm not seeing as many people maybe keep. I don't even hear about chillers really anymore in that, but maybe a lot of people are focusing on the popular fishes, and most of them tend to be a bit warmer, maybe. I don't know. But yeah. For axolotls, I don't see. So Stephen P has mentioned that chillers are popular for axolotls. I don't see that as much here, and they probably should do. I worked at a college. I taught animal management of exotics, and we had axolotls, and it was required to have a fan on them, which did keep them within range. But sometimes there was ice in the freezer, like in bottles to pop in, which I don't think is the best method. And the axolotls, I think, are maybe a little bit more popular than they should be, personally, given that there's plenty of other salamanders out there. I really like some tiger salamanders, and they although they do hide a lot, it may be a little bit more easier, I think, the terrestrial, or going terrestrial with anything is easier, personally. So on to research. So back onto, where to find papers. And a lot of people, I think it can be a little bit of a maze to find any reliable information, especially if you don't have academic access to papers. So there are repositories around. So this would be like ResearchGate, and I believe you only need to sign up with maybe where you went to school, so your college or school, I guess. Like school. American high school would be probably a better word. There's also academia, I think it's called, but I can't remember if you need institutional access to that or it just emails you, and it does spam you. There's another few of those. Sci-Hub is another I've never used. Usually by the time I guess it's on Sci-Hub, maybe I don't need. It's usually on somewhere else I can access. And there's a few journals that are really difficult to access, which would be like ichthyological exploration of fresh waters. Zoo taxa can be, but for a lot of these papers, scientists do put it on the repositories because that's what they do. And as Featherfin Film says, you can always email the authors. Authors are more than happy to send their papers. Usually there's a lot of sharing going around anyway, particularly from, if someone's got a particular interest in a group of fishes, they might actually have that paper on them. I have loads of papers just stored on my laptop anyway, and then I print out a load. So every paper I own ends up looking like a tip, because I literally, not I own, but I've printed out, because I put coffee on it or spill something on it, and then it ends up on my desk where it's there for months and becomes a pile that I have to put away. But here I've got the the revision of Corydoras, or Corydoridinae would be better to say than a revision of Corydoras. So I've got this, and a lot of people print them out and they're happy to take photographs of it if needed. But yes, it's all, people are more than happy to share papers. The only thing is definitely that critical analysis is needed. There's some crazy, there's all sorts of papers, and we can't forget there's been 2 actual scandals of scientific papers regarding Lancet Journal, which is a medical one. So definitely use critical analysis and look at the methods. Just quickly look at the methods, because some might be a bit off. So for the Lancet one was low sample size, and that's not rare. And sometimes, especially when you're using live animals, you will have some kind of limit to how much you can use, because there's ethics involved, and journals want those ethics, as well as they should be kept to anyway. So yes, definitely critical. I might do a video on critically analysing papers, but then I think half it is about learning on the way. And there's, when it comes to theses, they tend to be quite not too bad to access, but they tend to be on the university's website maybe, or shared publicly. Some aren't at all. So 2 of mine aren't. One of them, because it had no hope of publication, I put it on. I did a summary on the journal for the catfish study group, so technically it's kind of published. And then another I posted, which my Corydoras one was so useless, I think now I posted it on one of my Facebook groups. And then my ancestors one, I'm kind of holding it just in case, and then my coral one is open to the public for everyone to see how much I'm not very good with coral knowledge. Yeah, theses are everywhere. Just some people will even share their own. And also Google Scholar, a lot of people recommend it, and I don't think it's bad to actually find papers, but it doesn't mean there'll be open access. And I find it just as good to use search terms on Google. So I go on my Google search, I just literally put on the topic, like keywords, not even a sentence, and then put journal, and that usually brings up a lot. But there's a lot of secondary websites, like secondary research websites, reliable ones. So I've got one that I cite a lot on. So that's the scientific Fishkeeper.co.uk, I think I put it under. I should know my own website. Yes, the scientific Fishkeeper.co.uk. And then there's obviously like Fishkeeper and Chill, I think it is. Just checking the key. So that's run by a fish vet and a professional hobbyist with background in biology. Actually, no, she might be a biologist. And that's a really good website as well. But then there's also a lot of websites might be a little bit more of a community sort of contribution, like Planet Catfish. Scott Cat has loads of people write for them, and practical fish keeping as well. So there's a lot of good places to learn there. Obviously, there's some opinion things when it comes to like especially taxonomic, what species is what ideas. But definitely, and then like Seriously Fish, I think is really good. I don't think it's been updated for a while because some of it is a little bit out of date, to be honest. But it can be quite good. And a lot of these, if they cite, it's really useful for actually finding other sources. But But there's a lot of good websites, and most actually tend to be specialist, I think, because people are enthusiastic about the topic. But so for the films you mentioned, you miss Scientific Seriously Fish. Is it so I don't know, it might have had a community at some time, but it's been around maybe 20 years. So, and there were a lot of really good forms in the past. So Practical Fish Keeping had one. I believe the plant ones were particularly popular. Planet Catfish still has one that's quite good. Well, it's very good if you want to ever look at a form rather than using Facebook groups. And I didn't mention it here, but there are actually really good Facebook groups, especially there's a lot of scientific ones. So the main one would be like Catfishes of the World is very scientific, and that's one I tend to go on to. Loach Fanatics is pretty good. I don't know if they do post quite a few papers actually, and there's a few different like kerosene ones there, and then there's other ones which are more hobbyist sort of aimed. But there's quite a lot of like scientists on different Facebook groups, and Facebook groups get slated a lot, but they can be great places to find papers. And for a Facebook page that's very good, as mentioned, is Fish in the News. They have Facebook and Twitter. I don't really like Twitter too much, so I see their Facebook, but it comes up with the papers as they get published almost, and then an abstract, maybe a summary if the writer has access to it. But it's definitely worth liking their page and following it, just so you can keep an eye on the science. So I think it's a really good actual page for following things. And they also have a YouTube channel that I don't know how up to date it is, because I don't use YouTube as much as I used to. But the other thing I'm going to talk about is red flags of websites, because it's really difficult, especially when you're starting out, to know what is a good website and what isn't. And sometimes I'm a bit harsh with things. So in no particular order, I'm first going to mention the big one is incorrect identifications of the fish they have on the photos, which for new people, it might not be easy to notice. But over time, you might notice certain websites have a photo of a fish. Really bugs me when they don't cite the owner of the image. Plagiarism. If they're claiming to be a scientific website and they plagiarize, like, plagiarism is a massive thing in science. Anyone in science will have had endless talks, lectures, courses on plagiarism. So when people are not citing their sources, there is a sort of an exception for that. But definitely cite it. And AI has to be cited, because AI kind of actually counts as plagiarism. At least in science, you need to cite that you've used AI. And recently, I think there's been people going around Facebook groups asking questions and mining data, I guess. Or so I've been told, to then put on the website, which will really anger me if I see it. But that's a really common thing, though, just misidentifications in general. It's very common. People will post Hypostomus plecostomus, say common pleco, but post Pteroplectes, which is like Gibiceps, Padalis, Josomanus, any of them. And then that's not Hypostomus plecostomus. If they're saying this fish is Hypostomus plecostomus, then why are they writing a page about the common pleco in the first place? Because Hypostomus plecostomus is smaller than quite a few Pteroplectes. It's different entirely. And then also, where are they getting their research from, I guess? Another one is that I've seen is people posting Panax and saying Hypostomus plecostomus, or entirely different groups. It's quite common also for people to post other plecos as plecos that So where have they got that information to say that's what it is? And I guess it's common with other fishes as well. It's just I go straight to the pleco page, the Loricariid pages on websites just to double check, because that's where I judge first. But there's other things like Pangiocooli, bit of a hit and a miss one. But I think that's always worth mentioning, whether it is Pangio Mercy, Semicinta. With cichlids, are there? There's probably loads of cichlids. Corydoras, there's a lot of misidentification there. So if a website posts about a Corydoras julii, that is another sort of, definitely they might not be doing their research, because yes, there are Corydoras julii, but they probably should mention Corydoras. Now, I just realised I tripped up on a lot of that. Is it Hoplosoma now? Well, if they're posting Corydoras julii, they're out of date by about a week anyway, or a week and a half. Trilineata. I think they're lineage 9, which I'm probably totally out. Literally, I keep getting, yes, they are lineage 9, so they're Hoplosoma. Yes. So Hoplosoma trilineatum. So this is a weird thing with genera. When species change genera, genera have feminine, masculine, and neutral. So when you change the genera, it might have to change the suffix, so the end of the species name. So then you get it where it's changed. So trilineatus to trilineatum. Also, like what Megalichus doricata was Hoplosternum doricatum. It gets really confusing, and the Corydoras has been such a while, and I assume that what's the julii is the same. Just double checking, because I know my Corydoras so well. I really don't. Yeah, so julii is in lineage 9 as well. But when websites post, it's so common for them to get that mistake, and it's kind of basic one. If you're doing a care guide, I think it's much more important. If it's a general website just on general fish keeping, maybe not, but definitely if they're doing a care guide, get the fish identification correct first. And it's so common. Like there's I was looking at a website today because I'm after Gymnotus dravari, or any small of those very vivid Gymnotus. And I found a website that first, for misidentifications under knife fish, put that they're part of Notopteridae, which is an African group. And yes, African knife fish is, and I assume Aphas and clown knives, so are part of that group. The South American ones, Gymnotiforms, aren't even closely related. They're not even the same sort of clade. And the only fish they had available on that website was the South American black ghost knife fish, which is, I forget the species scientific name. Oh, names. When I'm ever on like a live video or something, I forget names constantly. And it's why for videos I have to just like do it again, and it stresses me out so much. Hate refilming videos because I hate editing videos. Yes, I assume it would be a Pterinotus albifrons, or would it be Pterinotus albifrons? It's one of those, I guess. If it's Greek or Latin, I can always ask my sister, I guess. But there's also the brown knife, which is also Pterinotus. But I see the name when I see the name, I recognise them. Like, that's it. But I did a whole article on on Gymnotiforms. That's the South American knife fishes, the true knife fishes, and a Pterinotus I thought was 2 species for some reason, and then I got and then I was like, actually reading the papers, there's quite a lot, and there's a lot of jaw diversity in them. And that's particularly interesting because I'm not sure how well it's been studied, how the jaw diversity of knife fishes might relate to their diet, because you do see a lot of diversity in what they feed on and how they feed. Pteronotus albifrons, common for good reason because it feeds so well, as does the brown one. Then in gymnotiform, the only other one I would say is gymnotus, and they can be either very slow. So mine, I have to either target feed in the right way because he won't compete with Neil the eel. Who is Macrognathus circumcintus eel, who eats a lot faster. So, or I have to cloud the tank. So I probably will separate them. I think Neil will be fine in that tank. So, what was it? Yeah, so red flags. I ranted about one for a while. So the other one is scientific names. I'm forgiving with it, but also if it's going to be a scientific website, scientists should have to use scientific names because they're universal. Common names vary so much. Like recently, so I think it was actually a year ago or 2 years ago, I was looking at a wholesale website. Why, I don't know. And they came up with whole new common names for L236, L129, I believe, as well, and loads of hypancistrus. All of the common names are like variants of zebra because zebra sells. And then I mentioned it, I was like, oh, look at this. Imagine if it catches on. And cracked line zebra, something like zebra, I don't know, wiggly line zebra caught on. I think L173 had one. Was that like false zebra? Which is really confusing because you've got the Colombian zebra, which is L1, Hypancistrus delbilitera. False zebra pleco, which usually gets called the false zebra. And that's really confusing, I think. So why that... and then people are using these common names. But if a website's going to do a care guide, please use a scientific name so we know exactly what species you're talking about, or use a genus name or something. But if it's a scientific website, why are they not using scientific names? Just because, especially if they're claiming to have a scientific background, they probably should. And if they're claiming to... I understand on Facebook and YouTube you can't use italics, but one of the things you're taught during your undergrad, if you're a scientist, is to put scientific names in italics. And we can forgive citations, but citations are nice. It's just I'm not innocent about forgetting citations for scientific names, because when you're writing an article, looking up the citation, then look up whether it needs to be in brackets, can be quite long. And I guess, yeah. But what helps with this is catalogue of fishes, solve scientific names immediately. Just put in the original name or the name you know, the scientific name, and it will come up with all the ones it's ever been. I think it did take it a few days to update for corridors. I hope it's been updated because otherwise I'd be using Planet Catfish for the names, and it's quite easy just to type in the whole genus and see everything that's ever been in that genus. And it's quite interesting. I put in Plecostomus, and the amount of species that come up for that. So Plecostomus is now Loricariid, which is in Loricariid, which is known as whiptail. So Plecostomus isn't what most people even call plecos anymore. So it's that's a bit of a mess. But what actually placed within Plecostomus was stuff like Ancistrus, so bristlenoses. There was 1 or 2 species in there. No Pteroplectes, so no common plecos were ever Plecostomus. Hypancistrus, I think, had 1. Quite a few Hypostomus, which are old school common plecos, I guess. So the last red flag for websites that are like scientific and so I'm reading red flags for scientific websites, so I'm not slating websites that don't, but this one really does annoy me, and this is standard length. So not everyone knows what standard length is. So standard length is, do I have an example fish on me? Not easy access. So standard length is tip of the head to the where the fin, the caudal fin, the tail fin starts. That is standard length. That's the measurement we use for most freshwater fishes, unless you're, I guess, going to weigh them like carp or something. And a lot of reliable websites use this. You might notice the initials SL, so on Planet Catfish, Seriously Fish, and then a lot of websites also use it but don't always make it clear. So Scott Cat does. And this measurement's really useful because it actually states how the fish are being measured. So a lot of websites, it will cause confusion because a lot of people will take a measurement as being total length. And if a website doesn't say how they're measuring the fish, how do you know what the total length is? So while it might not be a massive difference on like a neon tetra or a small species, on larger species, it makes a hell of a lot of difference. So take Panak even, where Panak, Shafari, one of the royal plecos, that's 60 centimetre standard length. And then it include the tail. That's probably another 20, no, don't know, 15, 20, then plus caudal extensions if they have one. And you're getting a much larger fish. And it probably is why a lot of people think their fish are gigantic or bigger than the measurements. It's just a lot of websites also copy other websites. So especially like I can imagine a lot copy Planet Catfish, Seriously Fish, and don't consider copying that how they're measuring the fish. So yes, as JT Fishkeeping says, measurements are always an issue. And the other issue with measurements is also like units. In science, we only use the metric system, which is a bit of an issue, I guess, sometimes in the UK because we use a whole mixture of different measurements. But also it tends to be millimeters when measuring fishes. So, and inches are so vague. You got 2.5 centimeters between each. So when you're saying, oh, it's 4 inches, it's quite a bit of give or take. So I see it as a red flag, but I'm forgiving on why people use it because they might not know. And going back onto the previous red flag, taxonomy does change a lot. As Feather Films has mentioned, cichlids are particular for it. We will always remember how big Cyclosoma was. And even as it, Crinocycla was much larger till maybe last year. Corydoras was quite a large genus of, 200 species? And now it is much smaller. I think it was maybe more, maybe 400. But I agree with JT Fishkeeping. Metric is it's way more useful because it is so precise. But it's understanding why people don't use it in a way. Some people might not. I don't know when. I think we switched in the UK maybe in the 80s. So there's generations which haven't been taught either. Yeah, barbs as well. I think, yes, is it tiger barbs that got moved or something? There's one, because were they Petra? No, they're not. I always have to double check these, and it's quite good to actually, I find taxonomic changes really useful because I double check and then I learn a little bit more. But yes, and then also it's quite useful to read the papers because what I didn't mention maybe about using where to research is that the scientific descriptions of the species are really good for actual ecological data. Many of them will record at least where the fish is caught and like how the habitat looks. Now the other ones decided to chew on their cardboard box, so they're going to be loud. But so what was I going to say? Yes, so scientific papers are really good for the descriptions, for the ecological data. Might even include conductivity of the water, temperature of the water. That's why I really like if I can't find an ecological paper, I'll go straight to looking at the scientific description. And some actually contain their dietary information. So for Loricariids, I think particularly like biologist has a really good one for the Xanthellus compared to Chrysolamus, and then also biologist dermatoides. So that's so gold nugget and mango magnum, and then also the green phantoms have quite a good comparison. And I wouldn't be surprised if Gara does get split up, as mentioned in the comments, because Gara is quite vast. So it has obviously traditional Gara flaviatus, which is the sort of the panda, the rainbow Gara. Then it's got the doctor fish, which is much more grey, and they don't really get the head growths of or the nodules. But what was it? I know the word, but they don't get those growths at the other. Actually, yes. So Gara flavitra, I think is that so rainbow panda Gara. They don't get the head growths of some of the other ones, tubercles. That's it. Which you do see in other cyprinids, other cypriniforms, particularly like goldfish, where it's quite useful to usually sex the males. They get these lovely tubercles on the pectoral fins around the gill cover. And it's somewhat similar to female Loricariids, where they get somewhat very light, almost like tubercles around them, the urogenital pore, genital pareil. So Featherfin Film says, Gara do get them when they're mature. It might be I've never seen a mature one. I've seen them get reasonably large, but maybe not actual mature one, because maybe I've never owned them. But when they come into stores, they're usually a reasonable size, because people don't want to see fully grown fishes half the time. Unless there's exceptions, but you don't want the old ones. It's really bad for, I think, for Loricariids, plecos, because where some get really large don toads, it just blurs their appearance. Like hypencistris just end up looking haggard with age. And then with like mine, the Baroncistris, they just end up losing those seams and the spots get smaller. So it's not really what people always want. Yeah, so Featherfin Film says he thinks these tubercles are an ancestral feature of cypriniforms and loaches lost. So that's really interesting, because it kind of brings up the topic of whether features are lost or whether they're gained multiple times, which logically, so we always mention something like parsimony, Occam's razor, the least assumptions is most likely to be true. So the least assumptions would be that it didn't evolve multiple times, and it potentially could be that ancestral feature of cypriniforms. And I always think of the example being like bats and echolocation, because I think some ancestral bats or some basal early branching bats had echolocation, but fruit bats don't or something. It's been a while. That was like undergrad, but cypriniforms are something I'm really interested in, but I've looked at particularly their phalangeal morphology. So they have, for those that don't know, cypriniforms have a very obvious phalangeal jaw. So that's that secondary pairs of jaws. And this is formed from the gills. From originally the gill rakers. So sometimes you see a little bit of retained morphology from that. So shape, and these are used to basically crush things. There's quite a lot of diversity in cypriniforms like carp and stuff of this morphology where they have teeth, but they've got very sort of, their phalangeal teeth compared to a lot of cichlids are like separate. So cichlids will have one unit, another unit, whereas cypriniforms are a little bit, they have two separate ones with quite large teeth, I think, but it depends on their diet. And cypriniform diets don't seem the best understood, because I did try and do some research on it. Especially carp, it seems a lot of people don't actually understand what carp feed on, other than they're omnivores. So there is like papers showing that they can and do benefit from those from the harder food items like snails, but a lot of people don't feed it to them. I don't know if it changed anything about it, but yes, they do feed on anything. I remember working for one store when they were trialing a diet. They used koi as an example of, these fish really love your fish food. And I said, I was like, well, it's in the back of the video as well. I was like, yeah, but they're carp, they eat anything. So not that I don't think they noticed. But yeah, so these secondary for jaws, cichlids are really well known for it. So they form what's known as labroids, with it's a polyphyletic name that is also connected with labrids. So labridae being what are they? The wrasses and stuff. But so what was I going to say? So they form that sort of group. So it's not labridae itself, it's labroids, I believe, is the name. So it's I'll put it down in the comments. But no one really uses this name. I just think it's a really useful name that appears in the or maybe it is to define labridae, but I've seen it used in the recent scientific literature, but it does look like the shorter name for labridae, which, so you are probably right, Featherfin Films on it, but they have these really thick phalangeal jaws, so this secondary pair of jaws that are highly diverse, and some are really good at crushing, some are really good at getting algae. There was another group of fishes, I think, that was in that group, but it's why, so cichlids are really well studied for it, but they have those very joined sort of strong ones, whereas cypriniforms don't. Although I am particularly interested in discus ones, there is a few images of discus phalangeal jaws, because discus are algae, orphuch, etc. specialists, where, well, they're not algae itself, but like what orphuch growing on like surfaces, so they're not really feeding on anything tough to digest. So I am particularly curious about it, and while I do have specimens, I don't want to take the jaws out because they're wilds. So Featherfin Films mentioned that there's new paper that I think this is the paper that moved a lot of families around. That's you're mentioning. So I think, is it Thomas Neer is one of the authors. I think it's built on a super tree as well. So I'm not entirely sure about it. I can't really say much about it. I've never used super trees, but then it is relying on those previous trees being correct, I guess. So I'm not really too sure about it, and I keep looking at it, and it also raises I've read it several times. It's just I think it raises what used to be more common, distochodids, like is it Distochodus sexfasciatus? Used to be really common, but now it's not so much. But raised them to, I think, family level. Loricariid got placed closer to cypriniforms, which has been suggested a few times. not cypriniforms, carassiforms. That's tetras, pacu, stuff like that. But yeah, so cichlids, I just put them in, perciforms. They're just this, it's a strange one because a lot of freshwater fishes are sort of in certain groups, and then you get, cichlids are just there in the middle. Well, far away from all the others. They're just weird, I guess, but there's quite a few of them. Yeah, so cichlids have been placed closer to Ovilentra. Cyprinodonta, which are closely related to cyprinodontiforms and silversides. So they've moved cyprinodontiforms quite a bit then, which I guess is another thing. So that sort of paper is quite difficult to look at the methods, because phylogenetics is a tricky one. So I was reading about super trees versus so when you build a family tree, so a phylogeny, it's loads of different methods, and if you've not got access to all of the data, or it's such a large tree, you need to, it kind of changes the topography, the shape of it quite a bit. So super trees, adding loads of trees together, whereas a super matrix is using loads of different, using the sequences directly. It takes a lot longer. So I've got one running, or should finish tomorrow. So that is 1,097 species from 5 different genes. So it's a super matrix, and that's taken 10 days. Previously, it took 2 weeks to run. And that's why phylogenetics, or working out family trees of fishes, is so painful, because literally I've got a sign on the computer saying, please don't log off, because I have to start again. But yeah, so taxonomy is quite understandable to be, like, I think if someone's got out-of-date taxonomy, it's not always the worst. Well, it's not the worst, especially if it's like the Corridora stuff is only recent. Corridora isn't a name before anyone ever complains about that. But if you want to shorten things, Hoplosoma's got an A at the end, so that will keep some people happy, I guess. And yes, so I was kind of just thinking about why. So for this, I kind of thought about, because of including science in fish keeping, biology and fish keeping, it's useful to think about all the different types of biology. So I've only included a few, and I was going to do really good case studies, but it's a lot more. Well, I can find papers if needed, and yes, Corridorus doesn't have a singular, it doesn't have a singular or plural. It's the same. It's just it's just Corridorus. I don't know where it came from, because it's not done with any other genre. I have heard people shorten Loricariid wrong. So people saying Loricaria, which don't do, because Loricariid is actually a genus, and the short, like Loricariid, so you'd say it's Loricariid, rather than Loricariidae as plural, which can get quite confusing. And I didn't realise that for quite a while, so I was trying to word the sentences around, say, Loricariidae. The other one is plural for genus, is genera, but that's just the word genus is genera, and it's quite common to make mistakes with that. And definitely no one says synodonti, which does sound kind of quite cool, but no. I don't know where. It might be one of those things that filtered through, because there are a lot of things that filter through, like Epistylus. That sort of peaked or it hasn't even peaked probably, it's just existed. That came from a certain website. With no citations on the page, I think, still. And every photo is of a different disease, I think, or pathogen. But that kind of, it's quite interesting to see how hobby information gets spread, and it sort of from incorrect sources. So some are definitely very good publicity. Some definitely know how to deal with social media really well. And that's how some things spread. And it's quite interesting. Like, for one thing, it never really spread that far. But there was a Facebook group that was saying, because, so for microscopes, you often need to use a stain. So you often need to use a stain to be able to see the microbes. Some are completely clear or just difficult to see under the microscope. So they took methylene blue, which is one of the stains doing this, to, if you put it in the water, if it's sort of, if the fish is highlighted, I guess, blue in places, that means that it's got a fungal disease. And that spread for a little bit. There's a lot of myths that spread. It's like plecos need to digest wood, which was originally came from Panac, and that lot need to digest wood, which came from Panac might be able to digest wood, and then the science proved it otherwise. But then it kind of spread to all of them eat wood at the same time, even though the science said in that time that they didn't. But it's interesting to see how things spread and what information spreads. And at different times, like the Woolstead has suddenly peaked again, whereas it kind of died down for a long time. And I actually have an old Practical Fish Keeping, which I never even noticed that it had an article, but it had a review of when the original book came out, which was quite interesting because they actually trialed it themselves. And there was like a good critique and for and against on it. So yeah, the website is, certain websites listed there, I will not say which. The dyed wood one, I don't know. I read the paper on. So there was the one where they fed the wood and losing weight, but maybe did they use dyed wood? That's the what's it? German 2011, and did they use dyed wood? There's a very interesting. I can link it. So he wrote for Practical Fish Keeping saying that loricariids don't eat wood, and interestingly, he got a load of hate from it. So loads of people really disliked it and wrote all sorts of things. So he then wrote on his website quite an interesting, detailed understanding of the topic, and he's not really a fish keeper either, so it's kind of from an outside perspective. Aquarium Delirium says he's scared to say hello just in case he gets wrong. There's no wrong, I guess, but there's always things to discuss, I think, as well. And what Stephen P mentions here is really interesting, because mentioning that Lake Vermont changes wildly in pH and quite rapidly. So pH doesn't matter for different fishes. So this is interesting because a lot of research and a lot of, when people talk about aquariums, they look at their own backyard, which happens to be usually the Northern Hemisphere in our case. So there's a horrific bias towards North American and European, also Australian ecosystems in understanding. But also as a hobby, we look at our local rivers and lakes, and they don't function the same. So it's it's really difficult because the science is there's real bias in the science. So that's it isn't as much science on neotropical, tropical ecosystems and their fishes. And I'm not so on going back to talk about wood and loricariids, plecos. Food companies adding oak into their food. So this has got more and more common, and I don't. So they usually say justified by science. I'm like, what science? But yeah, a lot of them do it, and sometimes I've seen the justification is it doesn't hurt them, which it doesn't. But it's a massive ingredient that has no purpose, so it's no different. Cereals, I think, have a small purpose, but it's no different from that sort of thing, or potatoes or something, and adding it in large amounts. Which is why I say that potassium morning wood isn't bad, but it's not the best. It's a bit of it's wasting a bit of money compared to if you got Soylent Green, Bottom Scratcher, Super Green, depending what needs it. And I think, yeah, it sounds good to a lot of people because now a lot of people believe that they need wood. And it's a really difficult argument that gets a lot of hate for some reason. People are very passionate about it. And this is a big thing for when I'm reading down articles on Plex. If they say needs wood for digestion or needs wood, eats wood, especially if it's not a panac or panaclus, I'll just be like, no. If it's a panac or panaclus, I might be like, I understand a little bit because they were thought to be. The others, nothing to do with it. Nothing at all. They just got added in. And so Aquarium Adventures mentions if it's a filler, no better than ash. So I talked to Nathan Hill about this because I was reading ash as well. And it looks like it says ash. So ash content actually refers to mineral content. So what they're meant to do is burn it all down and see how many minerals are in it, I believe. So it's not actually actual ash, which I managed to, so my video on it is probably not the best. But it's not all that bad. Even if they did add ash, at least it's got minerals to it. Like when you throw ash on a vegetable patch or somewhere with plants. But it depends on the source of it as well as how accessible is it, which I kind of added into one of my topics here on, well, kind of a lot kind of overlaps of diet because I find diet so interesting. But it is quite, fillers are interesting why they're added, but some of them aren't. The ones that really bug me is the cereal crops because they're cheap, and the potatoes, peas, and that lot because they're cheap. That's why the diets are cheaper, but it's understandable. And if you look at fishing, it's the same. And yeah, a lot of these wood diets are actually cellulose powder. Just saying. Which I don't know where people buy it for themselves, because a lot of these ingredients you could get from health food stores. So, as Featherfin Films mentions, there's a lot of reasons you don't see a gang of animals specialising in wood. It's really rare. I don't think there's any fishes that do. That's why there were so many studies on panac and panaculus, so royal plex, clown plex, flash plex, all of those. But yeah, so it's a bit of a weird, like, there is no fishes, but I really like the comparison. The author, I don't remember his first name, but German put up about diets of fishes and saying eating wood is like eating, is like parrotfishes eating sponge. They don't actually digest the sponge at all. They're looking for the bacteria inside the sponge, hence why they don't need the sponge. Hence why panacs are found in the Rio Xingu in Brazil without wood entirely. They just allows them to exploit a niche that others can't. So, and yes, the symbiotic bacteria. mean a lot. And there's really interesting studies on this. And I think I should have mentioned this in my developmental biology section, which doesn't sound the most exciting. But there's some interesting studies, or there's one particular one on discus, so Symphysodon, the genus, where they actually get a lot of their gut bacteria, like humans and mammals do from the milk of their parents, where discus actually get a lot of that gut biota. So it doesn't have to be bacteria. It's probably also like archaea and other groups. Actually get it from the mucus that the parents secrete. So it makes you think about how it might affect so how a lot of juveniles being raised entirely separate from their parents might actually affect them long term, because we know gut biota is so important. So I find it a particularly interesting topic, especially a lot of people are now thinking about it a lot, which is even just having other fish that have been in that tank before you add new fish, and also the waste, providing that gut biota to recover after shipping, which is something. Well, I mentioned more about fostering that sort of promoting the bacteria, but it's a really good concept, especially those of the same species that are doing well at it, having at least their waste in the tank before you add the new ones. Obviously, water change, keep the parameters good, or keep the parameters as low as possible in ammonia, nitrite, nitrate, etc. But yeah, so Loricariids, the ultimate detritivores, depending on the niche. Some are a little bit, and that's something I was, or potentially might mention, is how specialized are they, and which are they, sort of which species and stuff. Also comparing, I guess, the cichlids a little bit. And yes, so a lot of people with my project mention gape limited. So a lot of fishes are limited on what they can bite and chew and what they can feed on by the size of their mouth. So anything from most cichlids, carp, tetra. Obviously, you got ones that aren't entirely, because they can kind of break down the item, but a lot have to eat the item whole, and then they might break it down. Whereas Loricariids, plex, aren't as much, because they rasp, so they can break it down and then process it further. So they've already processed half of it. But it means they can exploit a niche that many other fishes would have to evolve quite far into. Maybe that's why Africa doesn't have as many, and maybe there are. Got a question. So on the wood topic again, oak bark being beneficial for stomach function. I don't think there's any research on that. So it'd be one of those things for research. You'd have to go down two avenues. I'm going to a bit, because otherwise I'm just only my eyes are showing. So I don't. So you'd have to. I can research it, and maybe if there's a way of like, I'll put any papers in the comments, but be looking at partially the benefits of oak themselves and whether they actually benefit fish. Problem is, there's also tannins can be a little bit more complicated, because tannins are anti-nutrients as well, which never gets mentioned, which means that they prevent nutrients being taken up, and it depends on the amount and the actual tannin. They're probably more likely to have certain antimicrobial effects, so maybe preventing certain of the bad ones, or the negative affecting ones. But a lot of these fishes don't eat oak in the wild. So Panak is particularly noted for, I think it's eating palm, which is nothing like most trees we have. And I did notice where I keep mostly loricariids, I do have panaculus. I did use a native what was it, beech or birch, and it literally grew so much bacteria, and none of the fishes would touch it, which was a surprise. The snails did, but it does show they kind of do partition that algae, bacteria, what they actually feed in terms of the microbes, because they touch none of it, absolutely none. And sometimes different ones of these woods will grow biofilms and they still won't touch it. And I can actually see what potentially might be a little bit of blackbeard algae, and they still ain't touching it. So there's a potential, but I can't really say. I can research it though and share any papers or ideas on oak itself, but other woods. I've looked into herbs, like basil has potential benefits, cumin, all of those, but there's obviously a sort of too much level, a sort of level of saturation and then a harmful level. Stephen P mentions for tannins, I assume cation exchange locking up micronutrients. I don't really know, and I don't think it's been that well studied, because for research, there's no real benefit for scientists really studying tannins, because it doesn't really affect aquaculture, really. Maybe if you're going to breed some species, but they're not ones that are going to earn a lot of money. The big ones are like discus, goldfish, sometimes like neotetra and stuff, but they need to be. So I'm not really sure. But I think there's no harm in having tannins, which is an entirely different subject, if those fishes are adapted to tannins in the water. So those Rio Negro fishes. But definitely not if you're keeping whitewater like mine. I've never probably seen tannins in their life. Well, apart from the captive bred ones. But yeah, so it's a difficult one. And there's been so much promotion of tannins that it's a maze to get through. So Titanicus, they're pseudocanthicus. I would always do minimum of 28 degrees for pseudocanthicus. They're generally found in those sort of habitats. Actually, that's a good. Nabokov, can you say how to actually make someone a mod? It would be quite useful, actually. But Titanicus, so the pseudocanthicus, minimum 28 degrees. You can go high, but I'd add in a lot more, increase the flow even more. They do want that amount of flow. They want a good high velocity water. They're riverine. At the end of the day, pseudocanthicus are quite adaptable. So water conditions, obviously low nitrates. Usually, clear water would be Nabokov. It doesn't seem to be showing. I can only delete or put people in timeout and stuff. So what I'll probably mention more. So on conditions, with pseudocanthicus in general, they're very highly territorial. So consider that with caves, and they can be from a young age. So I wouldn't keep them with smaller fishes. I wouldn't keep them with anything. Actually, that's because I'm using the wrong thing to look. 5 seconds. There we go. I was using the actual stream thing. There. So I've got 2 for now. Was it? So yes, just very highly territorial. Keep them in a way that there's not going to be too much competition for caves. Nothing too small with them. Nothing that they can actually trap. So similar sized fish or anything that's boisterous enough, but not too boisterous to feed. So they can be a little bit faster than most loricariids to feed, but they're not as fast as other fishes. So I'd limit it a little bit on the cichlids. So on most cichlids, it depends. Obviously, like discus and angelfish, stuff like that will be fine. You've got in your logo Clenocicla. Is it Severinus, your one? I think when I kept or worked with them, they were usually a little bit slower to feed, so they're usually fine. Hobbyists, the 2-power, I've never worked with, so I can't say how quick they are to feed, but generally not too many cichlids. The main thing, though, also would be diet. So for Pseudocanthicus in general, there's not the best records, but they are sort of more generalist carnivores. So feeding a lot more on a range of different invertebrates. So I'd feed them, yes. So just a mixture of different frozen foods, bottom scratcher. Avoid that fish meal. They don't eat fish in the wild. There's no records of any loricariids, any plecos ever eating fish in the wild. So that's an important thing, I think, to just consider. So, but Titanicus, I can't. There used to be a species that I always wanted, but then I realised I wouldn't have a big enough tank for quite a while. So I kind of forgot about them. And now Pseudocanthicus serratus is my favourite. So I'm just going to double check. So L273. Yeah. Serratus I worked with. Goth fish, totally black, beautiful things. Nasty as well. They had to be kept separately eventually because they, even at a young age, they get the white marks and mask on each other and in the store tank. So it's Rio Tapajos. But I can always double check and fact check myself and get some papers to double check the temperatures of the fishes where they're found in the wild. Especially because there's so many different rivers. But yeah, be a low conductivity river in general. So yeah, so for oak killing bad bacteria and possible parasites, potentially. It's just I don't think it's been studied at all. Oak specifically would be, it's not what is found in their natural environment. So if there is any certain toxins, they might not be adapted to it. And think about it, like palms and stuff like that have very different biology, I guess, to traditional. But yeah, it's something you could even experiment with it. And yeah, you mentioned, Mike M has mentioned his friend using oak bark to extract successfully when breeding tropical fishes. And it would, a lot of fishes people like breeding are, I guess quite a few are black water. And there you would probably get success. Other fishes, maybe it wouldn't make as much of a difference. The other thing is long-term breeding. So it's really noted with Hypancistrus, where they breed great for like the first 10 years and then they just stop and live for another 15, which doesn't make sense in fishes. And it does seem to be largely based on diet. So I think diet, because they're all feeding the same thing, and certain diets do seem to cause some infertility. But there's a lot more to, especially with certain fishes, and there's some fishes that have never been bred or rarely bred. And on diet, surprising how little there is on corridors. But a lot of the scientific descriptions, I went through a few. I could have gone through more. Didn't include diet, but they did include locality information, which I do want to look at Corridoridae a little bit more, but I need time because there is open access CT scans I can take a really good look at. But I need the time, and it depends what I do with them. And some of those open access scans aren't the clearest. So CT scans, computerized tomography lets me look at bones, lets me produce, which is the nice as well. They need gluing back, sorting out, but lets me produce 3D prints of fish bones, or just understand the fish bones because it's part of my research anyway. So I'll discuss some of the things that I've written down, because I think an important thing of understanding biology when it comes to fish keeping, particularly, is the background biology. So when I did my biology degree, I chose biology because I wanted the background to biology, because it will teach an overall of the subject. So first year I did all sorts of modules, and I even did plant biology till second year. Well, plant physiology, which is even worse, but I do like plant taxonomy. So it's something that we probably should go more into as a hobby, but there isn't always the people with the training or the background or the interest in it, like a real interest in chemistry, which I hate chemistry. But there's some really, I do recommend listening or reading the work of Dr. Tim Hovinack, I think it is. So Dr. Tim's, and he actually studied nitrifying microbes. So most being bacteria actually in freshwater, I believe, from the research, and the rest, so marine, there's a lot more of the contribution from archaea. But this really affects, I guess, how we keep fish when it comes to like bottles bacteria, how we're going to actually start the cycle, what we're going to use to start the cycle. Is there a difference between nitrifying bacteria? And temperature has a very little influence on the actual bacteria. So, and the background biology, so I really hate chemistry, so I read some of the papers and I'm like, but yeah, so that's the microbiology. The other thing is interesting about this sort of microbiology is actually helping understand disease, how it functions, how fish get it, what taxa are more likely, stuff like that. And so I'm going to get to the rest of the questions a bit later, but yes, organic chemistry, I did not do the best at it. I had to do a whole university module on chemistry in undergrad. Not fun. But chemistry does help understand water parameters. And I think ammonia, nitrate, we pretty clearly understand. There's the fish physiology element of it, which is more interesting. But I've written an entire article on that, so I don't have to talk about it because I read it and I'm like, all these different elements. But the big one is definitely when it comes like hardness. That is very complex in a way. I put it down further also because hardness comes into developmental biology, because carbonate hardness, calcium, etc., magnesium plays a role in how like bones are developed. Also, how porous the egg is, I guess I'll say. when they're laying eggs. It depends on obviously the wild habitat, but it's so it's really important. And I think there's actually a lot of gaps in the hobby to read. I think the chemistry particularly, but the harness is particularly interesting. Understanding how harness affects fishes, and it varies a lot on the fishes. There's adaptations to extremes. So even so Nebakov says, do I mean egg density when I'm talking about? I mean like the hardness of the membrane. So whether elements, compounds can pass through the egg. And it seems to be a particular issue with some soft water fishes where the eggs get so hard the fish can't actually get out of the egg. And they need help with like a paintbrush or something. I've seen it in Ancestors Dolopterus, Dolopterus, I prefer to say it. But hardness, yes, adaptations, even like neon tetras have a really extreme physiological adaptation to deal with that level of low pH, low hardness. Same for angelfish, but they can adapt to that mid-range. And it's whether that mid-range is harmful or not is, I don't think, the most understood, because why would you, when a lot of them are farmed in lower pH hardness ranges? And the one I kind of find more interesting than chemistry is geography. A lot of people do. I think it's not the best taught at school. When you do geography and talk about how rivers and lakes function, it's always, if you're in the UK, it's always horseshoe lakes, and just about that, I think, is what people remember. But they're so complex, rivers and lakes. Lakes, particularly the diversity of lakes. And how they function. I've done videos on it, which are particularly boring if people want to watch and learn about how rivers function, such as stream order, where how the rivers differ as you go down towards the sea or a lake. But that varies a lot. South American ecosystems, particularly. Or just tropical ecosystems in general. I'll just answer those questions before I forget. So yes, I do have CT scans of Pteroplectes gibiceps. I only did it kind of like by genera, unless the genus was particularly interesting, like Barioncystrus. I've got 2 of technically, because I wanted one of the Hemiancystrus, because I do also have one of Hemiancystrus medians. And also I've got 2 of Spectrocanthicus, 2 different Amirinus and Zooananoi, which is they're in a jar somewhere. The 2 Spectrocanthicus. And then what else did I double up? Hypancistris, I've got 3 different CT scans of different Hypancistris. I got Microcanthicus, which is Hypancistris Vandragitae, and also Inspector and Debita, I think, actually. Really tiny Debita. And so Mike has asked, do fish eat on certain things to help with mouth dexterity? I don't think they do anything to help with mouth dexterity, but there's certain food items that probably help that mouth dexterity if they're fed them. Like I think particularly, like you do see changes with hardness of the hardness of the food item. So I think using the muscles help develop the bone, sort of thing, where those muscles are being used if they're having to become more dexterous. Yes. So on to more interesting things rather than, I guess geography is quite interesting, but it is quite boring. So I think this is the one that has the most interest in Sorry. First, I think also Nabokov mentioned this pH drops, mentioning with eggs. I don't know, but I assume if it's too low, sometimes it might make the egg more fragile. But it depends on the fish. It might depend even on other things like the female's diet and stuff like that. There's so much that probably isn't studied because most of the studies will be on very hard water fishes kept in hard water systems. Well, not very, but you know what I mean. Not the ornamental fishes. So evolution, I think, is the most interesting. And I think a lot of people don't look at it as much. And the first one is the one that people actually do find boring, but I really enjoy, is phylogenetics. So phylogenetics is like the evolutionary family tree history of different organisms. So evolutionary placement, it helps a lot actually to know where fishes place against each other. I use it a lot in stores. If there's a fish I'm not seeing, but I know where it places, I can kind of work out a little bit about it, and then it gives me a little like boost without having to do a lot of research. But it doesn't have to be that intense. Just knowing vaguely that Hypencistrus is related to Picoltia, which is related to Scopincistrus, I guess. But Scopincistrus is a bit of an exception, I guess, to that group. But actually, a better example would be the Acanthus clade. So Acanthus clade includes Acanthus, so that's the Adenis pleco, also known as Acanthus adenis. Also contains Megaloncistrus, Pseudocanthus, I think. Oh, and Leprocanthus. These are Leprocanthus and Pseudocanthus, definite carnivores. Others, not too sure. No one's quite, doesn't seem. Megalocanthus, Megaloncistrus, potential carnivore specializing on sponges, which might be an interesting thing to research. But they're all nasty. Most of them get big, apart from Leprocanthus, which is still really nasty if they get mature. You could kind of predict a lot of things by that in a way. And then you can also, it helps predict care in a way and just diet and stuff like that. So the other part of evolution is what I kind of specialize in mostly, is ecomorphology. And so there's two aspects of it in here. Obviously cichlids, big topic for the most popular group for people to study in that context, but also like Loricariids is my speciality. But if you understand, and if you research and understand these, the morphology of the fish, you can understand like how what they eat, how they eat is really useful to actually know, because if you know how they eat, even without knowing what they eat, then you can kind of make an assumption. So long, few teeth, it might be more of a carnivore. And you kind of, I guess, loaches are quite, they've got that elongate jaw, a bit like Scopincistrus has that elongate, soft mouthpiece. There's a lot of assumptions you can make just by looking at how the fish eats or how the fish looks, and comparing to the scientific literature is really useful. But there's a lot more to this. So it's something known as the Liam's paradox. So fishes that, based on cichlids, fishes might specialize, but they are technically also well able to generalize. So this was based a lot on cichlids, I think Neolampologos or Lampologos sort of thing, where they might be algae eaters, they might be etc., but they're also feeding on fish given the chance. Obviously this was originally proven in captive environments, and fishes eat completely different things in captive environments to what they'd ever touch in the wild. but it was later proven in the wild. Problem is loricariids don't really do this because they don't feed on fish. There's no records of fishes in their gut. But there is some level, I think, especially the problem is with captive diets is it's the consistency or the actual part of it is no different. No different depending on what fish you're feeding. So there's a little bit less of anything you could prove on that. Mickey Emma's sorry, it's just I've got an article on this. I'm gonna probably got my website on the link, but it is a little bit on pH and KH just because it's quite a complicated topic. I hate discussing it in a way because it is a little bit more complicated, I think, than I like to discuss on video. But so yeah, so KH so I have dealt with but I worked entirely with a lot of soft water RO setups, and I used entirely I used the RO mixed to 100 ppm TDS, but I do water change a lot is the thing. So there's a lot more to it. I think some people might not be water changing as much, so there's that KH is being used up. They might also be, there's also gas bubble disease I experienced. There's a lot of people that I know that use pure RO and pure rainwater, but they also water change a lot. So I think it's a discussion that might need to be had. Especially when it comes to people on pure rainwater and pure RO that really that never had a crash. I got looked at like I was weird when I mentioned about pH crashes to some of them, but a lot of them breed a lot of fishes just on pure rainwater, but they might have a drip system or something, a lot of water changes. A lot of these sort of breeders do a lot of water changes. So it's a little bit of a difficult topic. And yes, as Stephen P says, it's a rabbit hole. I've spent hours on it, and it's the chemistry aspect. And also I think the problem is that when you're trying to research it, then you have to sift out so many aquarium websites out discussing, copying and pasting, which is why I don't like it as a topic. And wrong one. Mike mentioned Paracheedon. That's just so I'm getting the right genus because I always forget. Yes, Neontetras, cardinals, etc. So the blue colour, I don't know why they have it, and Sun and Dadio Axeroidy. I think they're one of the best nanofish. I never had an issue with them. I understand sometimes they're a bit sensitive, but I think they're gorgeous. So I think, so from my understanding, they're particularly Neon Tetra found in very blackwater habitats. So it might be, I believe it's something to do with, might be also to do with confusing predators, but Featherfin Film says that is to do with communication in the dark water. So in the black water. So it could be that. There could be, I don't know, has it ever been studied? I know, red coloration is easier to understand and counter shading, but my fish don't have the best color being catfishes. So they're a little bit less. But yeah, so discussing KH there. So the other thing I've put is just about developmental biology, because it's one of those major topics which really, I don't know if Sundadio axoides comes from blackwater habitat. I'm just assuming, although most deeper water fishes tend to be, or ones from dark habitats tend to be red for hiding. But actually, I don't know. Someone might be able to answer that, whether they actually come from blackwater habitat. But yeah, so developmental biology, so obviously done about eggs, bone development. A lot of people don't value this. So people talk about, especially when it comes to replenishing nutrients, is that fishes, snails, take up minerals from the water to varying degrees, depending on, some take a lot more up from their diet, some take a lot more up from the water, and there's a mixture of it. And why I put it in developmental biology, I don't know, because I've got my notepad. But this is one of the interesting things of science that I think there needs to be a lot more research on it, and especially the dietary contribution with where it's not, if the fish is an invertebrate, an insectivore, sort of something like that, it doesn't actually take up calcium or magnesium or phosphorus so well from the fish source that's in fish meals and most diets. But also the water contribution, because it gets often missed, and there's many other contributions from different minerals that are inhibitory or increase it and stuff like that. But so that's definitely something that probably needs thinking about. But that's all I had written down, because it was kind of, I didn't want to clash with people for the stream, but it's the only time I could do. Just because I don't like to go on too late, because it's nearly 10 o'clock and I don't like going past like 11, just in case not that I'll stay up that long. So if people have any questions, so I might talk a little bit. So with my tanks, I've literally got, I need to get more discus. I'm waiting till payday basically to get more discus, because I've got 2 males. I did try housing them together. 2 males does not work. Let's just say they do not appreciate each other's company. So I need 2. So ideally 2 females. If I could find 2 females, unlikely. So I'll probably put them as a one shoal and then let them pair off and work out from there. I had a dream about breeding Gymnotus drivare. So now I'm definitely looking for more of my zebra fish, so I can maybe breed them. I don't know why. It's just had a dream, and I was like, now I definitely want to breed them. And although in the dream I thought I had the dream, they were parthenogenetic, which means the fish was a female and just produced its own arm. Every time I do a stream, Neil the eel just tries to do something silly. So now he's trying to reach behind the fluval U4 filter, and I really hope he doesn't get stuck, because he's getting really fat now. Because he's the discus in there is on a diet, so he has a bit of that diet, and at least it's making him quite a bit big. And so Stephen P mentioned mineral diet of blackwater fishes, and I think that's actually an interesting thing to mention, because I don't think there's any research actually comparing their diets. Like, we've got gut records or wild records of what they eat in the wild, but we don't know what like any comparison really, because we can't really compare just off gut records for that sort of thing. But it would be interesting, because especially if there's something to do with anti-nutrients, maybe it might be there, maybe how they deal with it. I do actually have a textbook somewhere that mentioned it. I think it's neotropical physiology and ecology of fishes. It's probably not actually sure where I put it. Look, everything just ends up everywhere, like books on the floor all the time. But I would say maybe they definitely would need higher levels of calcium, but maybe our fish diets contain too much calcium anyway. A lot contain too much protein, I think. Especially the more protein, the more things that are wasted, the more that gets released as waste. Therefore, more ammonia, more nitrates. And some diets, it's really notable. So I think it's something that's worth looking. But yeah, so I know. So Stephen P mentioned dissolved organic content. I know there's something on that with isotopes as well, but I never looked at it compared to blackwater. There's a lot of blackwater Loricariid, but there's also a lot more whitewater ones, I think. And so protein, actually, I found, so Stephen P mentioned protein associated with high quality fish foods. So I actually found a paper. So originally, so if you want to grow a fish for food, it's really good to do high protein. You get a lot of high waste, but it makes them grow quicker. But then there's issues if you want to keep them long-term breeding, etc. But most of the science we have, and most diets are focused on that breeding and that growing for food aspect, not for breeding, not for growing the fish, not for colour. So that's why there's high protein, whereas it mentioned that lipids are more important if you're wanting to breed and want colour, etc. So yeah, it's to do with the most diets are based on if you want to eat the fish. And you'll notice it if you go to look at any food for like trout, etc. It will be like that. It will be the same sort of thing. Apart from some are really fancy. I found some, what was it? Banoffee, fish food for carp. Well, it's ground bait, but still fish food, I think. Yeah, so Mickey M mentioned has commented about why many showing fish are blue shimmering colour. I think it potentially does have a high contribution to why to predation. I think it might have some confusing aspect. Whether the fish can see, the predatory fish can see blue. The other thing is a lot of fish can see polarized light, which we can't at all. Whether that has an effect. There is a few papers on it. Well, if you look at the description of Corridors Bethany, that does mention coloration of corridors and how it might affect predation and stuff like that. But I don't know if it's the most studied. So it is an interesting topic. I don't do much coloration. I prefer, if it's something I can see, well, I see colour, obviously. Obviously, not everyone can. But I prefer like bones and stuff. I don't. A lot of my specimens don't have colour particularly well. Obviously, some do. So I don't know. It's impossible to know why some specimens hold colour and some don't. It's randomly got some goose feathers I found. But you can see this guy's got quite a bit of coloration, and there's fish that are dumped at the bottom because I ran out of jars. This one, for some reason, during preservation, I'm not even sure what that is. Maybe it is a tumour of some kind, but something's come out the gill of the pike there. This guy's kept colour. I only had him in formalin for probably only 3 days because I wanted to keep the colour. So this is a blue-eyed Panac cotyledon. This was donated to me, and this one is a little. What are you? Can't see from that angle. I think this is a little Ancestral Ranunculus, and you see he's held his colour, but I put him probably straight in ethanol because I'm testing out things. Whereas other things, it depends on the colour. I've got Platystachys behind there that still have all their colour, but I prefer not. And in the scientific papers, when you describe your species, most of them have lost all their colour when they're preserved, so I don't really use it. Yeah, there's quite a lot of mention of insects from above. There's a lot of popularity for like agapo fruit, which some fishes do use a lot. So that's like pacu, silver dollars, those large crassiforms that aren't carnivorous, or they usually are. Some, I mean, usually most of them will feed on a lot of like fruits and seeds and nuts. But it does, for Loricariids, they're not, some might feed on fruit, but it's difficult to say. Those would be like Hypancistrus. They do feed on seeds, where the seeds are from. The paper was very vague. It just said monocot or dicot, which is basically most plants. But it's quite interesting, especially, yes, the agapo side of things. Well, I don't own any Hypancistrus anymore, because I did breed them, but that was in the store context. I would say yes, better, and the licorice gramis parissimus. They definitely would feed from the surface. You just got to look at their, like, they have that superior mouth facing upwards, and also just they tend to inhabit that sort of realm. I've never looked at diets, but I would say so. Tetra, I think there's a lot more diverse in where they feed and what they feed on. In Blackwater, I think we also have to remember that fishes use more senses than sight. And whether they're just not even feeding on insects at all, really, or ones that are on a surface. So especially if they're on the surface, they're just going to be picking along, trying to find those, or waiting for movement that get picked up by the lateral line. Anyway, so I'm going to end this stream here because it's been over 2 hours. And if you have any questions, you can ask them in the comments, and I'll hopefully get back to them. Anyway, thank you for watching.